Hello students, in previous part we have studied about the different processes in human fertilization such like as oogenesis, spermatogenesis, menstruation cycle. In this part we will discuss on fertilization. Fertilization, a definition of fertilization is very simple. It means fusion of male gamete with female gamete to produce a diploid zygote. Fusion of male gametes with female gametes to produce a diploid zygote. This is called fertilization. Although the definition of fertilization is very simple, but the process of fertilization is very complex. How these processes can be completed? This process, fertilization process, completed in many steps. First of all, we can see that first process is approach of sperm. How the sperm enters into the vaginal part. We know that in one time ejaculation, the amount of semen is 3 to 3.5 ml. And ejaculation the value of or amount of semen is 3 to 3.5 ml, in which nearly 200 to 300 millions of sperm present. Why the sperm quantity is so high? This is because there are many barriers occurred in the pathway of the vaginal tract to pass out the sperm distance. How these factors? There are some factors which help the sperm to move in the vaginal pathway or reproductive tract, but there are some other factors which act as barrier. First of all, what is the approach of sperm? We know that in one time ejaculation, the amount of semen is 3 to 3.5 ml, in which 200 to 300 millions of sperm present. How these? What are the factors which affect or which help the movement of sperm? Help to sperm. Look, factors which help to sperm. First one is lashing movement of sperm or lashing movement of tail. A sperm move while it gets energy. We know that in middle piece, nipple mitochondria present which provide energy to the tail so that the tails move just like a spiral, lashing movement shown by the tail. So the sperm moves in. Second one is prostaglandin. Prostaglandin, a chemical substance. With the help of prostaglandins, this chemical substance help in contraction and relaxation of the uterus as well as vaginal pathway, and sperm moves in reverse direction. It means while it produces contraction in vaginal and uterus pathway, due to this contraction the sperm moves upper side, and so that is called a reverse direction. Prostaglandin, due to this chemical contraction can reduce in vaginal or uterus path or uterus tract. So that sperm moves in reverse direction, it means upper side. And third one is aspiratory movement of the uterus. Aspiratory movement of uterus. Uterus contract and it provides reverse direction the sperm movement, that is called aspiratory movement of uterus. These are the factors which help to move the sperm in the reproductive tract in female. But there are some other factors which affect or which act as barrier while the sperm passes through the female reproductive tract, and those are barriers which create obstacle the movement of sperm, which provide obstacle to the movement of sperm in a reproductive tract. First one, the vaginal pathway is acidic in nature, and in acidic in nature, sperm degenerate. First of all, we can say that acidic pH of a reproductive tract. Third one, in vaginal pathway, the cells act as phagocytic. Those phagocytic cells degenerate the sperm. So we can say that phagocytosis. Phagocytosis process is produced in female reproductive tract. This is the second obstacle. And third one is, as we know that as the ovum is released through the ovary, the endometrium, uterus inner wall start to thicken, and as the cell moves through the fallopian tubes and reaches to the uterus, at that time thickness of endometrium will be high. This high thickness of endometrium create obstacle to stop the pathway of sperm. The third one is thickness of endometrium, inner wall of uterus. We have seen that just like this. This is the uterus. As the ovulation process started, this uterus inner wall start to make thick layer of endometrium, just like this one. This thickness of endometrium create obstacle, create problem to pass the sperm through this pathway. Sperm move from this one to different directions. This may create problems. So these are the factors which affect the pathway, which act as barrier in the pathway of reproductive tract. One more thing is, in this approach of sperm is capacitation. Capacitation. What is capacitation? Attaining the maturity by the sperm in the reproductive tract of female, that is called capacitation. To attain functional maturity by the sperm in female reproductive tract. sperm gain functional maturity in the reproductive tract in female, that is called capacitation. This capacitation process is completed in two ways. What are those ways? First one is removal of sterol or glycoprotein from acrosome. The first process is removal of sterol or glycoprotein from acrosome, and second one is taking in taken in calcium ions. How this process can take place? As we have seen that in a sperm, head structure contains this one like, and this outer membrane is called plasma membrane. Plasma membrane. Just inside the plasma membrane head, a cap-like structure is present here, and this cap-like structure is called acrosome. This acrosome contains sperm lysine and enzyme. At the head part or cap-like structure plasma membrane, it contains many glycoprotein or sterol substances, just like this one. These are the glycoprotein particles. These glycoproteins are removed with the help of some chemical substances which present in vaginal pathway. In vaginal pathway, suppose this is the some chemical substances. The chemical substance which is present in vaginal pathway. This chemical substance remove this glycoprotein, just like this one, and they attach here. Due to removal of glycoprotein, the plasma membrane becomes very thin. It means it is very permeable and it becomes very thin, so that acrosome may burst out. First process of capacitation is removal of sterol or glycoprotein. As we know that at the cap or just around the acrosome in plasma membrane there are many glycoprotein present. The chemical substance which is present in female vaginal pathway, this chemical substance degenerate the glycoprotein, or we can say removal of the glycoprotein from the plasma membrane, and these glycoprotein are attached to this chemical substance, so that the plasma membrane becomes very weak and it acts as permeability. It provides permeability so that the acrosome may be burst out. This is the first process. And second one is taken in calcium ions. As the plasma membranes become weak, through this plasma membrane, calcium ions enters into the sperm. As the plasma membrane become thin, the calcium ions enters into the sperm. Due to the entry of calcium into the sperm, this provides very active. It means it makes the sperm very active, and sperm show vigorously movement. Again, listen, what is capacitation? Capacitation is the process in which a sperm gains functional maturity in female vaginal tract, female reproductive tract. This process is completed in two steps. First one is removal of glycoprotein, and second one is taken in calcium ions. How the removal of glycoproteins can take place? In female vaginal pathway, some chemical substance present. This chemical substance remove the glycoprotein which present in the plasma membrane. Due to the removal of glycoprotein, the plasma membrane becomes weak and act as permeability. provide permeability. It means entry of any substances. This process called removal of steroid glycol. As the plasma membrane becomes thin or provide permeability, the calcium ions enters through this edge. Calcium ions enters, this calcium ions makes the sperm very active, so that sperm show very vigorously move. This is the first process which is known as approach of sperm. The second step is fertilizing or anti-fertilizing. During fertilizing process, there are many steps occur. First process is approach of sperm, how the factors help the passing sperms and what are the various steps. And second one is fertilizing or anti-fertilizing. Reaction. It is not a reaction, it is only identification of ovum with sperm. Fertilizing is fertilizing chemical substance which is produced by ovum. Anti-fertilizing is produced by sperm. The chemical substance which is produced by ovum is known as fertilizing. It is also called spermophilic site. It means the site from where the sperms enter into the ovum. Just like this one. Suppose this is the membrane of ovum, and just outside the membrane, ovum releases a chemical substance that is called fertilizing or spermophilic site. Just this is the spermophilic site. A spermophilic site is present in fertilizing maybe 2 or 3. This is the site where the sperm attached with the fertilizing. Suppose here the sperm is attached. So that sperm enters through this fertilizing and then enters into the ovum. This is the second process which acts in fertilizing or anti-fertilizing. Fertilizing is the site from where the sperms attach and enters into the ovum. Anti-fertilizing is produced by the sperm. As we have seen that in one ejaculation there are nearly 200 to 300 millions of sperms are ejaculated or released, but only 100 sperms are reached in fallopian tube. And out of this fallopian tube, out of these 100 sperms, one fertilizing site contains 2 or 3 sites of attachment of fertilizing or sperm. There are 2 to 3 fertilization sites. There are 2 to 3 fertilization sites at ovum. And secondly, we have already studied sperm always enters into the ovum through animal pole. It means the pole which is near to the nucleus. So, if an ovum contains 2 or 3 fertilizing, this is the fertilization, and fertilizing contains 1 or 2 or 3 sites of sperm, attachment of sperm. So, there are only 10 or 15 sperms are attached to the fertilization out of 100. 200 to 300 millions of sperms are ejaculating one time. 100 reaches to the fallopian tube. 10 or 15 sperms show attachment with the fertilization. What is the process occurred during this? First one is it maintains the uniqueness in the species. It means the adult one, or we can say the young ones, are produced of the same species. To maintain the uniqueness of the species. It maintains the uniqueness of the species. This is the second process of fertilization. And third process of fertilization is degeneration of Graphian follicles, or we can say that burst out of sperm lysine. Third process. The third process is breakdown of sperm lysine. Sperm lysine is an enzyme. It is an enzyme. It contains three enzymes. First of all, the sperm lysine contains three enzymes, and those three enzymes are: first one is corona penetrating enzyme. We also can say that a corona digesting enzyme. Corona digesting enzyme. Second enzyme is hyaluronidase. Hyaluronidase enzyme. And third enzyme is acrosine. Sperm lysine contains three enzymes, and those three enzymes are: first one is corona penetrating or corona digesting enzyme, second one is hyaluronidase enzyme, and third one is acrosine. As we have studied the diagram of an ovum, just outside the wall of ovum, it may contain many corona radiata cells, just like this one. Corona radiated cell, it digested with the help of the enzyme that is called corona penetrating or corona digesting enzyme. This enzyme to digest the corona radiata that is called corona digesting enzyme. The corona radiated cell, these are made up of follicular cells. These cells are attached with a special chemical substance, chemical acid, and this acid is called hyaluronidase enzyme or hyaluronic acid. This is hyaluronic acid. Again, corona radiata cells are made up of follicular cells. These cells attach with a glue, and that glue acts as a hyaluronic acid. It means these cells are attached with each other, just like a glue-like substance, and that glue-like substance is hyaluronic acid. This hyaluronic acid is digested by the hyaluronidase enzyme. So we can say that it digests the hyaluronic acid. So there is leakage can be take place in the corona radiata cell. And after that, acrosine. What is the role of acrosine? We have seen that the sperms or ovum is contained with the vitelline membrane. Just outside the vitelline membrane is zona pellucida, and after that corona radiata. Corona radiata is digested with the help of these enzymes. What is the role of acrosine? This acrosine digests the zona pellucida. It means acrosine, it digests the zona pellucida. The middle layer of the ovum, which is made up of jelly-like substance, and zona pellucida is made up of the secretory material of the follicular cell. Follicular cell itself makes the coronary data, and the secretion makes the zona pellucida just inside the vitelline membrane. Vitelline membrane is made up of vitol. It means yolk-like substance. Yolk. So these cells are breached down and they make just this one like substance. It means the decomposition digestion of the coronary data cell. Always again listen, sperms enters into the ovum through the animal pore. Just like it, this one is the nucleus. This is the nucleus. The side which is closer to the nucleus, that is called animal pore. This one is animal pore. So all these sperm lysine enzyme act towards the animal pore so that it can digest the coronary data, zona pellucida, and it remains only vitelline membrane. Just like it, this one vitelline membrane which is made up of its own cell. This is the vitelline membrane. So there are three steps occurred. First one is approaches of sperm. Second one is fertilizing or anti-fertilizing reaction. It means identification. Third one is breakdown of sperm lysine. After that, sperm lysine, the next step is formation of reception cone or fertilization cone. Number 4. First process, approach of sperm. Second is formation of fertilizing and anti-fertilizing reaction. Third one is breakdown of sperm lysine, and fourth one is formation of receptor or receptor cone. Receptor cone is also called fertilization cone. We have seen in this process coronary data, zona pellucida is breakdown, so it remains only vitelline membrane. Just like this one, it remains the many coronary data cells are breakdown. The site from where the sperm is enters into the ovum. This pole is called animal pole. Towards the animal pole, the vital suppose this is the sperm's breakdown of acrosome. And as the sperm reaches near to the vitelline membrane, the vitelline membrane shows just a projections or conical structure towards the sperm, so that the sperms can attach with this cone, and this protrude part is called receptor cone, as well as we can say that fertilization cone. As the sperm without acrosome, this is near to the vitelline membrane. We have seen that in this part, digestion of coronary data and zona pellucida. The innermost membrane is called vitelline membrane. As the sperm reaches near to the vitelline membrane, at that time vitelline membrane show some projections, some protrude part, some conical structures to attach the sperm here. This protrude or projected part is known as the receptor cone. In next part we will discuss on some other processes which take place in the fertilization process.